Hey, good evening, and welcome to the 2019 Cell Biology Exam Review with BioLerner, Ms. Lerner. Happy to see you all here, or maybe not see you, but interact with you online here. I'm going to pause for a little bit and just let people catch up with me. Hey, Liana, good to see you. Awesome. Okay, so, normally I just wait for some questions from all of you. Hopefully you have many questions. I don't really have anything special to say except to, do your best to just focus on the basics, make good connections, use all your good strategies, and hopefully everything will go as you would like. Good evening. Thanks for joining us, Telmetrics. Always happy to see you. Hi, Ankita. Okay, so I think people are with me now, so go ahead and start to chime in with some questions. Hi, Raga. Okay, while I'm waiting, I will do my usual kitty introduction. So this is Pinky. I think you met him the last time. Hopefully the other one will come out this evening as well. We shall see. Okay. Hi, Kush. Hi, Namik. Okay, we have our first question coming in from Namik. How does light affect the rate of transpiration? Yeah, it's funny, Namik, nobody asked about that this year with the transpiration lab. Maybe we just went too fast or something. But this one is a little strange because in terms of the actual rate of evaporation, light doesn't really make things evaporate faster, right? Heat does. But light is a trigger for stomata to open because, of course, there has to be light present for the plant to do photosynthesis. So if there isn't any light, the stomata are going to tend to close in a plant, right, in order to conserve water. So it's not so much that light affects the actual rate of transpiration, it's just that you're more likely to have the stomata be open and therefore have more transpiration occur if there is light. So I would say it's more of a trigger for a plant to open its stomata than that it directly affects the rate of transpiration. Adrian, hello. What role do plant pigments play in photosynthesis? Okay, so that has to do with the idea that certain kinds of molecules can absorb certain wavelengths of light energy. And many plants, of course, are green in color, which means they're reflecting green and absorbing other wavelengths of light. But there are many pigments actually in plants. I mean, if you just think of the colors of different fruits and vegetables, right? I mean, there's many, many pigments in plants. Some of the pigments that are not green can also be useful for photosynthesis. I'm thinking of a particular example, which is that sometimes when there's a plant with its leaves near the ground, it's actually able to capture sort of reflected light, so the light hits the ground, and these are these very tiny low plants on the ground and bounces up off the ground, and a lot of times those plants will be more kind of a reddish color, and that will allow it to kind of absorb that low light a little bit better. And so that's an example. But plants use pigments for a number of things, just like we do. So sometimes it is for other organisms to see, right? But there are pigments other than chlorophyll that can allow plants to do photosynthesis. Ankita is asking, for the plant diagram, for cellular respiration, should glycolysis be included as well? I don't think it's really specifically needed. The model is a little bit more big picture. I mean, there are details that are asked for in the prompts, and I meant to actually get out a copy of that before I talk to you guys. I might reach over and grab that in just a second, maybe when the cat's not on my lap, so that I can see the prompts while I'm talking to you. I don't think that level of detail is necessary. No, we really want to tie it in to the idea of, plants have to do respiration so that they can use the food they make to produce ATP, which gives them energy to live and grow and build molecules and everything else that they need to do. So the cellular respiration ties in that way. The only reason I taught about glycolysis was to help you to see the differences between fermentation and respiration, which is not really particularly relevant to plants. Plants don't do fermentation that I know of. So I can't think at the moment of any reason you would need to put glycolysis on your plant diagram. So Kush is asking about osmotic equilibrium versus dynamic equilibrium. Okay, so dynamic equilibrium is a much more general term. Okay, that can be used in any situation where things are still happening, but there's sort of no net change. Okay, so it's a sort of idea of stability, right? Sometimes you have in chemistry a chemical equation that's molecules are moving in both directions at the same time, and it's sort of I try not to use the word balance. It's one of these kind of not helpful words, but it's it's a relatively stable situation. So that's a general situation. Osmotic equilibrium refers specifically to having this equilibrium of osmotic pressure. So the main example we used was, actually I'm going to revise that. So an osmotic equilibrium, I think in the purest sense, would be like an isotonic solution around something, where you have as much water moving across the membrane in each direction as the other. And so I would call that osmotic equilibrium. And I think the implication is that you have the equal concentrations of solute, or an isotonic solution. I think, though, that you're thinking of the example of the plant cell, and that's not truly osmotic equilibrium. It's really sort of it's more of a dynamic equilibrium between two sort of forms of pressure that make up water potential. So you've got the sort of osmotic pressure, and I may not be using that word technically accurately, but you've got the sort of osmotic pressure of the water, fresh water, tending to move towards the solutes inside the plant cell, and then the sort of equal and opposing pressure of the water pushing from the inside, turgor pressure, up against the cell wall. And when those forces come into equilibrium, right, the plant cell doesn't necessarily change much. Individual molecules of water could still be going in. It's not in osmotic equilibrium, because if it's in fresh water, you still have a much higher concentration of solutes inside of the plant, inside of the cell, I should say, than in the fresh water. So it's never going to reach osmotic equilibrium if it's sitting in fresh water, because it's never going to have fresh water inside, right? But it is at dynamic equilibrium because those two forces are more or less balanced out. Okay, questions are starting to come in, so I'm going to try to speed it up a little bit here. Mar was asking, when the ADP and the phosphate bind in ATP synthesis, does the enzyme connect them together through a chemical reaction? Is that written right? I think she's asking. Just one second. Yes, there is an enzyme that does that, Marmo. You didn't need to learn the name of it, although it's kind of intuitive. It's called ATP synthase, and it's one of the ways that ATP is made. If you want to learn more about it, the website where you learned about ATP on learn.genetics does a really nice job of sort of showing a graphic of that. But honestly, for our purposes, we're not really going into that level of detail. So if you just sort of get the idea that this ATP synthase is making the ATP in the mitochondria, or in some prokaryotes without a mitochondria, it is a chemical reaction, essentially. Yes, energy is being transferred and bonds are being formed. So yes, that's a chemical reaction. Joseph is asking, does both photosynthesis, I think he means, and cellular respiration build ATP? Yes. Okay. I didn't even go into that in class, and I don't want to confuse anybody. But remember, there are a lot of details to photosynthesis and cellular respiration that we didn't cover in my class, Miss Lerner's classes. Okay, some of the other classes went into the detail of sort of the intermediate reactions of photosynthesis and cellular respiration. It's one of those times you get to be grateful that I'm actually covering less than the other teachers, okay? But there is a part in the middle of photosynthesis where ATP is sort of acting as an energy carrier, okay? You don't need to worry about that. The net result is that you're forming glucose, okay? You don't need to worry about what happens in between, okay? The net result of cellular respiration is that you're forming ATP, and that's good for now. Okay, Liana mentioned something about telophase and anaphase. Okay, again, that's another area where that is not required in my class. Okay, trying to let go of some of that detail. You guys have enough to learn. Okay, so you do not need to know the phases of mitosis. You do need to know the phases of the cell cycle, and people have been struggling with that a lot. So the cell cycle includes interphase and cell division, right? Cell division, of course, includes mitosis and cytokinesis. But you don't need to know the individual phases of mitosis. Sahib is asking about cell cycle regulation. So, in terms of what can happen if the cell cycle isn't regulated, um, it could be a number of things. It could be that that particular cell will divide, but things will go wrong and it will be damaged. It could be that that cell will become damaged enough that it will self-destruct in apoptosis. If that is not able to happen, then that cell could become cancerous. And, all those things are possibilities when the cell cycle is not being regulated. Ankita's asking, do specialized cells have the same genes? Yes, they do. We're going to hit that a bunch in genetics. They have to have the same genes. They all came from the same fertilized egg. Every cell in your body has the same genetic information. Okay? What it does with it is very different. And I will use the analogy in genetics a lot of a cookbook, so I'm just going to start that now. It's like we both have a big fat cookbook full of recipes, and you're making chocolate chip cookies, and I'm making chicken soup. Except I don't make chicken soup, but you get the analogy, right? We all have the recipes for everything. We don't all make the same ones, and specifically, our cells don't all make the same recipes from our big cookbook. Is transpiration the same as osmosis? I'm not sure who that is, but I'm happy to answer your question. Not at all. Okay, so transpiration is the evaporation of water out of the openings in leaves, called stomata. Okay, so that's water literally evaporating through a hole into the air. Okay, osmosis is water crossing a membrane from generally one solution to another. Okay, it could happen through an artificial membrane in a lab. Okay, in our class, we mostly talk about it happening through the membranes of cells. So maybe it's water is moving from a guard cell to the surrounding cells, right? Or from the bloodstream into a cell, or from the cell into a bloodstream based on the osmotic balance. Okay, so osmosis is specifically water moving through a membrane. It doesn't involve any evaporation. Okay, let's see. Oh, thank you, Adrian. I should read ahead. Sometimes you guys answer each other's questions better than me. Okay. Yeah, thanks, Joseph. I'll be using that terminology around genes being switched on and off a lot during genetics. Okay. Lucas is asking the different parts of a chromosome and when do they split into the other parts? I'm not sure I entirely get your question. I think you're talking about the chromatids. I'm not sure, so clarify if that's not what you're talking about. But the idea is that a chromosome itself is DNA wrapped around proteins, and that we'll talk about that more in the next unit, but it wraps up so tightly that you can almost start to think of it as just being this one chunky thing, okay? And that chunky thing, which is like millions and millions and millions of nucleotides wrapped around proteins, if it needs to replicate, it makes a copy right next to it, an identical copy, a sister chromatid, attached in the middle at the centromere. Okay, when that cell divides, or at least when the nucleus divides in mitosis, those two chromatids are pulled apart, and each of the daughter cells gets one of those chromatids and has the same sort of chunk of genetic information that the original cell had. Okay, I'm going to leave it at that. Caleb's asking, when we run, if we burn through our ATP, do we switch to fermentation? Okay, I'm going to clarify that. We're never going to burn through all our ATP. Okay, if you went a second without ATP, your cells would die and you would die. Okay, so that doesn't happen. I think what you're talking about is, well, you might be talking about that. So you start doing, first of all, you're always doing a little bit of fermentation, okay? If you started to run, you would run out of the ATP in your cell within seconds. So immediately your cells are going to ramp up and start doing fermentation, okay? As soon as the level of, I don't want to say start, doing more fermentation. As soon as the level of ATP starts to drop, there's actually another fallback, something called creatine phosphate that we didn't talk about that can spit out some ATP really quickly as well. But at any rate, very, very quickly, within seconds after you start to run, the muscle cells are going to start ramping up fermentation and pumping out some more ATP that way. okay? And I don't know the mechanism, but what I think happens is that after another, 30, 40 seconds goes by and you're able to increase the level of cellular respiration because you've had time for the oxygen to get to your cells because you're breathing faster and it's increasing the oxygen concentration in your blood, and that's increasing the oxygen diffusing into your cells. As your cells increase the level of cellular respiration, I suspect there's some feedback mechanism to decrease the level of fermentation. I don't know exactly how that works, but that would be what I think would happen. Okay, Ankita's asking about energy flow for the plant diagram. Okay, so energy flow means the same thing it's meant since the very first week of school. Okay, you might want to look at that stuff from the first week or two of school, right? What did energy flow mean? Think about your hand in the water. Okay, so the idea is that from the first week of school, we talked about how we could trace energy from its ultimate source, which for most life on Earth is the sun, through living things in the process of photosynthesis and cellular respiration since the very first week of school. So all we're doing now is we're sort of filling in some of those details, but I want to make sure you're connecting back to that big idea. So I should be able to see on your model where the energy's going, right? And maybe it's outside, maybe it's in a molecule, maybe it gets moved to a different molecule. You show me where the energy's going and where it ultimately ends up, okay? Okay, Adrian's asking about cell division. I can briefly hit them, although I'm going to recommend that for a really basic question like that, there's a slide right in the cell division debrief, right? So do remember to use your resources for that. But if you think about sort of why your cells would need to divide, certainly if you're going to grow it all, your cells need to divide. If you're going to repair, like you need to build new skin or something like that, you're going to need to have cells divide, right? The one that people tend to forget about is that if you're a unicellular organism, like an amoeba or a bacterium, then your cells are going to divide simply to reproduce, right? To go from one organism to two. Okay, so that's called asexual reproduction. Okay, glad I answered your question, Lucas. Okay, hold on, I'm going to scroll through a few things here. Okay, and Keach is asking about apoptosis and cell cycle regulation are not caused by mutations. They are, but if there are errors and mutations, then cancer is caused. Okay, so when we talk about the word errors there, I shouldn't say we're always talking about mutations. So I think you're getting a little bit of that cause and effect mixed up. So hopefully you wrote down that diagram I did in class when I handed back the development in cancer papers. Go ahead and go back to that. So I know it can be a little confusing because we use a general word like error sometimes in more than one way. So for example, I said errors in DNA replication. That means literally putting the wrong nucleotide in the sequence of DNA. Okay? And that error, mistake, is called a mutation. A mutation is simply a change in the DNA. Okay, there could be other errors, like maybe the chromosomes don't pull apart properly, maybe they both pull to one side or something like that. So error is just sort of a general word for mistake. Okay, not all errors, some errors cause mutations, some cause other problems. Okay, so you do need to have mutations in these genes that help to regulate cell division and that allow cells to undergo apoptosis in both of those things in order for a cell to become cancerous. And this is going to be multiple mutations because there's not just one gene that controls each of those things. There's going to be many genes. So there have to be enough mutations to cause both of those processes not to work right in order to have cancer. Caitlin's asking about differences between alcoholic fermentation and lactic acid fermentation. The sort of obvious difference from their names are the products, right? You start with the same reactant. You're starting with pyruvic acid either way, okay, which has energy in it, right? And if it really has to do with having enzymes that cause different chemical reactions to occur. And this is really just kind of an evolutionary difference. Over time, some organisms evolved a pathway where they had enzymes to break down the pyruvic acid into alcohol and carbon dioxide, so organisms like yeast, right? And other organisms had different enzymes and broke down the pyruvic acid differently. So if you're a lactobacillus bacteria or an animal muscle cell, you can break down pyruvic acid in a different way and end up with the product of lactic acid, right? So they're just two alternative pathways for being able to continue to do fermentation without oxygen available. Yes, Adrian is trying to clarify, do we only need to know what each phase generally is for the cell cycle? Yes. So as long as you understand the basic ideas that interphase is sort of normal cell function, if the cell is going to divide in the sort of not too distant future, then interphase would also include replicating the DNA, growing, but lots of cells also just hang out for quite a while, right? And they just don't divide at all. And I don't know, I can't remember right now if it was in the materials I gave you, but sometimes that's called G0, and the cell sort of almost goes out of the whole cell cycle and just kind of hangs out and is a cell for a while, right? Sometimes it continues kind of through that cycle, right? And starts to go through the steps that will allow cell division to occur. Okay, as far as cell division, you just need to know that it's mitosis, how the chromosomes actually divide in the nucleus, and not the individual phases, and the idea of cytokinesis, that the cell itself has to actually break in. The cell membrane has to be separated and make two new cells. Ankita's asking about, do blood stem cells only produce red and white blood cells and platelets? Yeah, there are a bunch of types of white blood cells. We're not going to get into that, but you end up with kind of a number of cells that can be produced. But those are the main names that we would use, would be red blood cells, white blood cells, and platelets. Yeah, thanks, Joseph, for clarifying about the different organisms performing the different types of fermentation. How does the humidity increase the rate of water loss during transpiration? So there's a couple ways to think about that. First of all, just as a reminder, nice little review. Okay, low humidity is dry. High humidity is more moist, more moisture in the air. And the way you can think about that is it's really a concentration difference. If I have something that's really wet and something that's really dry, that's kind of like a big concentration difference of water, right? So the water will evaporate more quickly, or transpiration would occur more quickly when you have the plant, which is generally wet on the inside, right? And the air being very, very dry. Okay, if the air is more moist, those two areas are closer, and you're not going to get this evaporation or transpiration happening quite as quickly. Okay, so on a moist day, the transpiration rate is lower, and that's going to allow the plants potentially to still do the same amount of photosynthesis. If they still have light, it's fine, right? So that they can still do plenty of photosynthesis and keep their stomata open while losing less water in transpiration. So that can be a good time for plants to do photosynthesis when it's kind of humid, maybe in the morning, right? The light is good, but they don't lose quite so much water in transpiration. Okay, Joseph's adding a little information about differentiation in the blood cells. Nice work, Joseph. Okay, so he used a couple fancy words there. Progenitors, kind of like a precursor, but sort of the original cell. Precursor we use more for molecules and chemical reactions. Progenitor is more like a cell that gives rise to other cells. Okay, I'll just throw out there, because it'll come up later in the year, these blood stem cells are often called, and I'll use the word pretty soon in class, hemopoietic stem cells, the ones that make these blood cells. Adrian's asking how ATP is connected to plant structure. Well, let's see. I'm going to refer you back to the cactus writing, okay? And I'm going to remind you that structure means what things are made of, right? So if plants have to build structure, you should know what that means. Check your cactus writing, okay? Reminder that structure consists of having the molecules themselves, but that you also need ATP to build molecules. So remember that one of the main uses of ATP is to be used to build molecules like polymers. Okay, you can't just put them together all by themselves. You've got to add some energy to that. Okay, so that you want to think about this point I keep hitting about plants, that they have so much glucose that they make in photosynthesis, but they have to use glucose for many different purposes. They're going to build glucose into all their other molecules, like amino acids. They're going to use some glucose in cellular respiration. They're going to store some glucose for the future in starch. They're going to have to build cellulose to build their cell walls, right? Many of these uses also require ATP to build the molecules. So some of the cellular respiration is going to be for ATP to keep the cells alive, whatever, maybe do active transport or something, right? Some of the ATP is going to be used to build other molecules, some of which will end up being part of the plant's structure. Okay, Leonie's asking again about the two types of fermentation. So I did a fairly lengthy explanation of that. So if you have a more specific question, let me know. Okay. Okay, sorry, I fell behind here, so I'm going to read a few things and then I'll get back to you guys. Wow, okay, I fell right a little far behind, so I'm going to read a bit and come back. I'm smiling because one of you said, according to Google. Google isn't really a person, you know that, right? Okay, can you explain the process of enzymes building up monomers into polymers? Um, so that's a kind of chemical reaction. It was actually on some slide presentation. I think it was the one about proteins and enzymes that showed a particular kind of chemical reaction called dehydration synthesis. You don't actually really need to know that. You just need the basic idea that organisms build monomers into polymers, right, in order to build these bigger structures, and that process does require some energy in the form of ATP. Okay, Krish asked a question about the cytoskeleton. Cytoskeleton is actually kind of more complicated than we went into. It does a number of different things. Sometimes it's actually providing like physical structure, sort of, you know, giving the cell a certain shape, because remember the outside of the cell is actually pretty gooey. Sometimes it's those little tracks on the inside that you saw the little motor proteins walking on. So there's actually different parts of the cytoskeleton doing slightly different things. Okay. Okay, good. I like how somebody, I think it was this person, hi, that I don't know, they were clarifying that the stomata are not cells. Yeah, and that's important. The actual cells are the guard cells on either side. The opening in the middle is a stoma. It's not a cell, right? So that's why you can't have something like osmosis happening into the stomata, because there's no cell there. There's no solution there. Mar was asking, do we need to know detail about leukemia and blood transport? I'm not sure what you mean by blood transport. No, not specifically. I mean, there was some information about leukemia and blood cancer just to kind of give you some context, but no, you don't need to know about that specifically. Okay, big long debate about what diffusion is. You don't have to have a membrane for diffusion, okay? You have to have a membrane for osmosis. Diffusion can happen in the air, okay? It could just be molecules moving through the air. It can happen in liquid. You don't need a membrane at all. Okay, Katie, sit down. Okay, Laura, I think I already answered your question about connecting biomolecules to plant growth. Okay, Neha's asking a question about the equation of cellular respiration. Not sure you necessarily need to write the equation. Not sure. You could probably show it. But at any rate, if you want to write it, that's fine. I think it does help to clarify that you understand it. You absolutely do not include ATP in the equation. The way you have it written in your question, Neha, you're saying that glucose and oxygen somehow is building ATP. Okay, there's no way that glucose and oxygen can build ATP. That's C6H12O6 plus O2. How are you going to get ATP, which has 3 phosphates and a nitrogen base, out of glucose and oxygen? Okay, so if people write that, I think you don't really understand how molecules work, right? Because in a chemical equation, you're just rearranging the atoms in a different form. You can't poof, make all new atoms appear, right? So the ATP is a different reaction, right? And I've written it on the board several times, the idea that ADP and phosphate are using energy from the reaction where glucose is broken down in order to build ATP. So refer back to your notes to see a better way to write that. Okay, Marwa, no, you don't need to know about in great detail about that kind of stuff. I can't remember. I may have some reference to bone marrow or something on the test. I don't even remember, but it would be much more general. Okay, so that was from the stem cells thing, and you know that was one of those assignments toward the end, which was kind of more just for general interest. That stuff's in the news a lot. We're going to be going into genetics, so I'm not particularly. I don't have anything about cord blood or anything like that. That was partly just to expose you to some interesting ideas that you might hear about and that we'll touch on in our next unit. And the way you would know that is that those things are not listed on your unit guide at all. So, you know, remember that that's a good reference, not just for what you do need to know, but sometimes for what you don't need to know. Okay, Ankita says, so cell cycle regulation and apoptosis are mutations? No, they're processes. The processes are directed by genes. Okay, genes have the information for those processes to occur. So if those genes have mutations, those processes might not occur correctly. Okay, so if you have mutations in the genes that allow your cells to regulate the cell cycle and allow your cells to perform apoptosis when needed, then those processes won't happen, and then you could get the uncontrolled cell division of cancer. Liana's asking about NADH and NAD+. Okay, so again, if you're in Ms. Lerner's classes, you don't need to know about that level of detail. I will tell you, though, I do have a resource for you, okay? If you would like to see me, Ms. Lerner, okay, in a BioLerner video showing those internal processes of photosynthesis and cellular respiration. I don't do it in the way the other teachers do with the eddy electron. I can't keep track of that, okay? But I do it with some sort of, I don't remember, I have these manipulatives on the board, like little pictures of molecules, and I move them around the board and show all the stuff about NADH and electron carriers and all that stuff. Okay, if you would like to see that, look on my BioLerner channel, and there are some videos for photosynthesis and cellular respiration, and I sort of act all that out on the board. And you might find that helpful if you're in one of the other teachers' classes and you need to know that detail. Okay, so feel free to check that out, but I'm not going to go through it in the live stream because my students don't need to know that. Ryan is asking, is the Krebs cycle aerobic? Again, you don't need to know that for my class, okay? The electron transport chain is where the oxygen is required. I'm not going to say any more than that, okay? It's the very last step. For our purposes, we're sort of summarizing all of that as the part of cellular respiration that happens in the mitochondria, and that is aerobic, okay? We call it aerobic respiration. Okay, Eric, I'm not sure who you were responding to. You said the process of converting glucose into pyruvic acid. That is the one level of detail I covered, that is glycolysis. Again, I really only covered that to connect to fermentation. That's really the only context you need to know it in. Okay, ooh, I think something here. I'm not sure what's going on. There's a comment here from Liana saying chromosome is condensed and the other is uncondensed. I think you're talking about chromosome and chromatid, but that is not the case. Okay? Chromatid means a single. Hold on, the cat is stuck on my sweater. Okay. So earlier I talked about the idea of sort of one chromosome being sort of this continuous chunk of DNA wrapped around protein, and that when the cell does DNA replication, it makes a sister chromatid. Okay? One is not more condensed than the other. All chromosomes condense during the time of cell division. Okay? It is what makes them into this sort of solid chunk that we can see under a microscope. Because if they didn't do that, they're sort of all loose, like a bowl of noodles, right? The analogy I use sometimes is that when the chromosomes are uncondensed, less condensed, that they're like a bowl of noodles, okay? So that they're actually, your cells can get to them. When the cells are going to divide, you wouldn't want to try to divide a bowl of noodles. They'd get all tangled and broken, okay? So what happens is like those long noodles sort of scrunch up together more like a bowl of rice. And then you can kind of easily move them around and separate things without them getting all tangled, okay? So condensing is something that happens during the time of cell division. Most of the time in your cells during interphase, your chromosomes are less condensed, okay? But that is not the difference between chromosomes and chromatids. Kayla's asking about advantages to using proteins as fuel. There aren't really a lot, right? Generally, the only time we use proteins as fuel, or at least in any great number, is going to be if we're starving and we're mostly out of our fat and our um, carbohydrate reserves. Okay, mostly there's disadvantages. A, that we don't have very much. We have to break down our organs to get it for the most part. And also that it creates a great amount of these sort of nitrogen byproducts that your body has to excrete because they build up and become toxic potentially. So it's really more disadvantages, and that's why we don't use it very much. Adrian's asking about plants getting water from the soil. It kind of works the same way as the guard cells, right? So this was in your original How Plants Work. The idea that the plants, if they want to sort of facilitate the water moving in, they can actively transport ions from the soil into the um, root cells, and then the water will follow by osmosis. Okay. Sometimes I hear people say the roots suck up water. Ooh, roots are not sucking up water. Okay. So the water is flowing in by osmosis, and once it's inside of the plant and makes it to a xylem, then it will be sort of through transpirational pull drawn up into the upper parts where the leaves are and go out through evaporation. How come the rate of evaporation is fast in rainforests if there is high humidity? Minji, I'm not quite sure. I agree. I think it should be relatively slow, except that there is a lot of water around, but I'm not sure where you're getting this idea that the rate of evaporation is fast. Um, Maybe you can give me a little more context for that question. So I'm kind of figuring out where I am again here. Okay, I see a bunch of people answering the question about chromatids and chromosomes. Thank you. Okay, Samir is asking about how does glucose help to build up amino acids. So the way you phrased your question is unclear. Okay, you're not building amino acids, like out of glucose, right? I mean, it is a different molecule, but organisms can use enzymes to change one molecule into a different type of molecule. So for plants, of course, they can only make glucose through photosynthesis. Okay, so unless it's some ion they're getting from the soil or water, they're making everything else. So way back, we talked around about the idea that a plant has the enzymes to convert a glucose molecule, add in some nitrogen, okay, in a certain form, and be able to create amino acids that way. But again, the glucose isn't really building up amino acids. It's not like amino acids are made of glucose, right? But it is providing the raw material, some carbon, hydrogen, oxygen, that the plant can use to form amino acids through different chemical reactions. Okay, a bunch of questions about ATP synthase. That's simply the enzyme that forms ATP. Okay, and ATP synthesis just means producing, building. So that's just the building of ATP. So ATP synthase is used for ATP synthesis, hence the name. Marwa asked a specific question about the test. There's a fair amount of multiple choice. There's some matching, a couple of matching sections. I think that's all. I'm trying to remember out of my head. I think it's mostly multiple choice, couple matching sections, and then the model. Okay, Adrian asked a question about insulin and blood glucose. I'm going to see if somebody responded to that one. Question about stem cells. So I'm going to read Daniel's response here first, and I can give feedback on that. So Daniel said insulin is released into the bloodstream by the pancreas. Receptors on the outside of cells pick up these insulin proteins to perform exocytosis and add more protein transporters. Yeah, you've got the right idea, Daniel. It's that last part is phrased a little in a way that might be difficult for people to understand. Cells don't really pick up insulin proteins. The insulin binds to the receptor on the outside of the cell, and it just stays there. And while it's bound to the receptor, that triggers those glucose transporters to go from being an internal vesicle and then sort of through an exocytosis kind of a movement become part of the outer cell membrane, allowing glucose to move through the transporters, facilitated diffusion. Okay, when insulin comes off the receptor, those transporters merge back into an internal vesicle, and the glucose uptake is going to stop, or at least that form of it. Okay, thanks, Daniel, for helping to answer some questions here. So on the stem cell one, you wrote cells that have a wider future range. Yes, more. The word we use for it is potential. Okay, just like all of you have the potential to do so many things in your lives, and you will. It's pretty awesome. These stem cells have the potential to be many different kinds of cells in their future, right? And then as differentiation goes on and the cells become more specialized, that potential sort of reduces, right? It's sort of like when you major in something in college and then you have a little bit less potential to get different kinds of degrees, right? You're going to get like one kind of degree. So, you sort of become over time a little bit more narrow in your range of possibilities. Okay, hold on. Cat issue. Give me one moment here. Okay, yeah, thank you, Cat. Sorry, Cat climbing someplace he wasn't supposed to. Okay, let's see. Laura's asking, I'm skipping some things that are either you guys already answered, I've already talked about. So there's one here by Laura that says, does glycolysis break down free fatty acids? No, glycolysis is a specific series of reactions that breaks down glucose and forms excuse me, pyruvic acid. So for free fatty acids, if you actually look on, I don't know if it's on your metabolic pathways drawing, that one may not be advanced enough. There's a whole series of these reactions that go from glucose to form the ATP in cellular respiration, and the free fatty acids sort of join in later on, past the point of pyruvic acid. Okay, that's a little hard to explain with words. But the answer to your question is no. Glycolysis is specifically breaking down glucose into pyruvic acid. Let's see. Okay, hi Leo. Water goes into the water through the roots. Okay, so let me clarify something, Leo. Leo's asking about water going into the roots, and he's mentioning the potassium ions. The potassium ions was in the context of the guard cells. It's not like that's the only ion that plants can move, okay? So there's a lot of ions and solutes in the soil, and the plants might move different things in, right? It doesn't have to be potassium ions. Water will follow different kinds of solutes. It doesn't have to be potassium ions, okay? Mar was asking whether plant cells can perform fermentation. I don't know of any examples of that. I don't know that it's impossible, but I don't know of any examples of that. Plants have a lower need for ATP in the first place, but all organisms I know of that can do fermentation are either bacteria or various kinds of eukaryotic cells. I haven't heard of plant cells doing fermentation. I don't want to say that for 100% sure, but I don't think so. Chris is asking more about fermentation. I'm going to be honest, Chris, you don't really need to know that much more than what we did in class. The main thing to know about fermentation is to understand that cells can do things without oxygen, right? They can still produce ATP, and that there are these different pathways by which cells can produce ATP from their food. And some of the advantages and disadvantages of that. That's really all. Of course, you can learn a lot more about it, but you don't really need to right now. Neha's asking, do organelles like chloroplasts and mitochondria have their own membranes? Yes, they do. And in fact, what's interesting about them is they have a double membrane. And the reason for that is because they evolved from prokaryotic cells. So, I know I use my hands a lot, but see if we can make this work. Here's one prokaryotic cell, right? It has its own membrane around the outside. Okay? This other cell, a different kind called an archaea, does exocytosis and wraps that in a vesicle, which is a membrane. So now, and it pulls it inside the cell. Now around that prokaryote is actually two cell membranes. Its original one and the one that formed around it during endocytosis. Okay? And that's one of the important pieces of evidence that helps us to understand that organelles came, or at least organelles like the mitochondria and the chloroplast, came from prokaryotic organisms, is because they actually have two separate membranes, one from their original membrane, one from the endocytosis event. G is asking about humans only use lactic acid fermentation. I assume you're comparing that to alcoholic fermentation. Internally, yes. Sometimes this jumps around. Give me a moment. So Ankita's asking, she's saying chloroplasts and mitochondria have their own organelles. No, they don't. Chloroplasts and mitochondria are organelles. They don't have organelles because they evolved from prokaryotic cells, and prokaryotic cells don't have their own organelles. Those organelles exist because, at least mitochondria and the chloroplast, because another cell took in the prokaryote. Ooh, I'm going to take advantage of this. So this is my other kitty. I don't think you've seen her yet. Her name is Penny. She's very pretty. She's a pastel calico. She's a little shy. Okay, relax. It's okay. She just wants to sit on my lap. She doesn't really like to be in the public eye. Okay. So let's see. There's a bunch of questions going around about the ions taken in the roots, blah, blah, blah. You should know the general concept of water following the solutes, and that's how organisms move water, for sure. Other than that, I don't remember. I don't think it's on the exam specifically. I kind of focused a lot more on the guard cells, so I wouldn't stress out about it. But you should know the general concept of moving solutes in order to make the water move. Okay, so some questions about sort of differentiation and specialization. Um, yeah, and Leo, that's kind of the way I think about it, is that differentiation is the process of a cell becoming specialized. But when I went back to look at the wording, the video does call the specialized cells differentiated. I know it's a little confusing when people use these words in different ways. It's not a big deal. I think just for clarity, to realize there is a process, they don't just poof become specialized, right? That this is kind of a thing that happens over time. So I like the word differentiation for that, because specialization you wouldn't really use for the process. That just is kind of a reference to having specialized cells. So that's why I like the word differentiation, but it's not really wrong to say that specialized cells are differentiated. That's just another way that the word is used. Liana, you're asking a question about if a cell gets too big. Again, that's actually something I didn't cover a lot. It has to do with the sort of cell division part, the idea that that's part of the reason cells divide, because there's disadvantages to cells becoming very large, right? It has to do with surface area to volume. I didn't cover that. But you can imagine if a cell is bigger, just having things diffuse across is just a big hassle. There's just a bunch of problems with cells becoming sort of too big. And so the solution to that in life is that cells stay small and they just divide and make more of them. So your textbook does a decent job of explaining that if you want to know more. Leo's asking about the plant absorbing sunlight. Should we have a chloroplast? I think if I wanted to know how plants grow, I would definitely need a chloroplast. Yeah, that's a good thing to have. Question about, is cholesterol itself a hormone or just a precursor? A precursor, as far as I know. Cholesterol, hormone is like a messenger. Cholesterol doesn't really act as a messenger. It is a lipid, has a number of roles actually. The main role it does directly that you guys have learned about is the fact that it helps to stabilize animal cell membranes. But it's not really sending messages. Okay, Ankita's asking, so for cellular respiration, one of the products is energy. You see it written this way occasionally. I really am against the idea of calling energy a product. Okay, energy is more like a state, right? Or there are certain amounts of it, but it's not a product in the way that molecules are. You can't rearrange the atoms of it, right? So I really want to separate in your minds the idea of the molecules and the energy, or it just gets way too confusing. Okay, so energy is not a form of ATP. ATP is essentially a molecule that helps to carry energy for us, just like electricity in our houses. Okay, so the products of cellular respiration are carbon dioxide and water. Okay, energy released in those reactions is used to build ATP. There's a question about cancer. Hold on, I just lost it. Okay, so G was asking if someone has cancer and their cancer cells continue to divide, do those cells just stay in there or will they eventually die? I'm not sure what those cells are. I assume you mean the cancer cells. Part of the problem with cancer cells is they don't really die very easily. They just keep on dividing. That's kind of the problem. So they tend to kill the other cells around them. Then over time, they tend to sort of move to other parts of your body and start to build new colonies of cancer cells, right? We call that metastasis or metastatic cancer, or malignant. You hear that term. So, yeah, that's the problem. No, they don't really die. Um, somebody named Arna, hello, is asking about differences between plant and animal mitosis. I think the basic process is the same. There might be differences, but it would be way beyond the scope of biology class. The cytokinesis part is quite different between plants and animals. And again, I didn't go to that level in my classes, but animal cells pinch together like this, plant cells stay their same general shape, and they build a new cell wall down the middle. So that part is quite different, and you can see a picture of that in your textbook. But again, you don't really need to know that for my classes. And we'll go over the practice exam in class tomorrow, so don't worry about that too much right now. Okay, so Alar's asking about the question on the review questions about muscle cells storing and using energy. Okay, so you want to connect that to a whole bunch of things that we've learned, right? That in terms of storage, the only molecule that muscles can store is glycogen, right? But to store glycogen, they have to get glucose, so you can connect that to insulin, as we talked about earlier, in terms of glucose uptake. They also have, once they have the glycogen and break that down for glucose, they are some of those rare cells in their body that can do either lactic acid fermentation or cellular respiration. So they're sort of a nice example of both storage and use of energy in different ways. So that was sort of the intent of giving you that example, was to allow you to put together some of those different concepts. Leon is asking, do closed stomata completely stop water from flowing out of the cell? Okay, so I'm going to change your language. It will be from flowing out of the plant, because no water's flowing out of a cell. Stomata is just a hole, okay? But the ultimate answer to your question is no. Plants can still lose water from their regular cells. It's hard to stop any water at all from evaporating, like they're not that sealed up. Plants do tend to have sort of a waxy surface to try to reduce the amount of transpiration, but there is a little bit of evaporation that happens even when the stomata are closed. Okay, Temme wanted to clarify about the guard cells absorbing water to close or to open, okay? So the guard cells are going to absorb more water. That's going to increase the turgor pressure, which, because of their already sort of being shaped like little kidney beans, when you increase the turgor pressure, it actually makes them bow outward and open the stomata. When the water leaves the guard cells, following the solutes, then the turgor pressure kind of reduces. They sort of collapse inward and close the stomata. Stoma, if there's only one. Okay, Marwa is asking if the test will have written questions. So it's going to have a model, right? That could have a little bit of writing, but remember, you're going to really show through diagrams and pictures that you understand what's happening. Adrian's asking, is cholesterol a steroid hormone? No. To clarify that again, cholesterol is not a hormone. It's used as a precursor in other chemical reactions to make steroid hormones. Okay, I've already answered about the protein, so I'm going to let that go. Do all animals perform lactic acid fermentation, or is it only humans? My impression is it's animals in general. I'm not going to say that's all animals because, that's a lot of different things on Earth. But, I think lots of animals can perform lactic acid fermentation in their muscle cells and maybe in a few other cells. Marwa, do we have to draw sunlight being used for photosynthesis? Well, I told you to trace the energy flow. So what do you think? You think you need sun to show energy flow? I think so. Does the rate of transpiration refer to the rate of water loss? Yes, essentially. Because remember, in the context of plants, transpiration just means evaporation. I'm not sure if that term covers just through the stomata or through other parts of the leaves. Actually, I'm not sure. But yeah, for our purposes, pretty much. Adrian is asking, what is a steroid? So steroid is a chemistry term for a group of molecules that share certain characteristics, okay? Just like alcohols are a term, right? There's a bunch of molecules that because of their chemical structure are called alcohols. Or, a bunch of molecules that because of their chemical structure are called amino acids, right? So there's a group of molecules that because of their structure, the kinds of atoms and the shapes they make, are called steroids. And they typically have like a ring and some fatty acids or something. I don't know, I'd have to look at some more pictures. And it so happens that some of the hormones in our body are made of this type of molecule. And so they're called steroid hormones. A lipid with a 4-ring structure. Okay, that's fancy. I'm not sure the 4-ring part. 4? That sounds like a lot of rings. Maybe so. I haven't looked at a picture of one in a while. Leona, acetyl-CoA is another one of these intermediaries in the Krebs cycle that I didn't cover in my class. Again, check out those other videos. Kankita's asking about sister chromatids during interphase. The sister chromatids don't exist until after the DNA is replicated. Now, that does happen during interphase. So if the cell is going to divide and it goes through the S phase, it will build a sister chromatid, and those will exist until mitosis occurs and they divide into 2 parts of the cell. Okay. Yeah, Laura, it was funny. I'm not sure I've really thought about that idea before of what the cancer cells do in interphase, but it seems reasonable to me that they wouldn't spend a ton of time in interphase. They're going to have to still go through that phase because they still have to replicate their DNA and grow in order to divide. But yeah, there's no real reason for them to sort of hang out long, and they do divide pretty rapidly. So I imagine that they tend to spend less time in interphase, certainly than your average cell that's kind of hanging out in interphase a bit more, doing its regular function. It was really, that question was really just to get you thinking about the more rapid rate of the cell cycle in cancer cells compared with non-cancerous cells. Okay, Joseph was asking a question about something I mean in the interphase. I'm not sure I can trace this question back. There was kind of a discussion going about the phases of interphase, the parts of interphase. Joseph was saying there's some phrase in the textbooks, but other resources say it's G1. I'm not sure. Off the top of my head, there's G1, the S phase, and G2 that have to do when it's growing and all that. Again, though, to be honest, I'm not really focusing on that very much. You should have a general idea of what happens in interphase. That's enough. Ananya, hello. What is DNA overload? That I cut that out this year. I'm constantly trying to cut stuff out, believe it or not. It's in the textbook. It has to do with the idea that um, if there's too much activity going on in the cell, or if the cell is too big, that it just can't get to the information in the DNA fast enough all at one time. Okay, and that problem is called DNA overload. It's one of the reasons why cells can't get too big. Yeah, that's about it. That's more than you would need to know for my class. Question about what mitosis actually is. Technically, it is the division of the chromosomes in the nucleus. Okay, and the nucleus itself. So, you have one nuclear membrane with chromosomes that dissolves, the chromosomes divide, and each one forms a new nuclear membrane. So you go from one nucleus to two nuclei. Okay, a little bit of a question going on about cellular respiration and the amount of ATP. Okay, so Marwa, now you're saying yay, only multiple choice, but I did say there was matching. Multiple matching, beware, multiple matching. People struggle with that. Be very careful, think carefully. Multiple matching. Okay. Um, Leo's asking about breaking down fatty acids. I think this came up a little bit ago, but fatty acids only go through cellular respiration that I'm aware of. I don't know of a process in which fermentation breaks down fatty acids, only glucose. So Laura's asking, do muscle cells only do lactic acid fermentation, alcoholic fermentation? Only lactic acid fermentation. We don't do alcoholic fermentation. That would be way too convenient for us. Okay? People drink enough alcohol. They don't need to be doing that. Yeah, to clarify, Leo, about cellular respiration. Glycolysis is the first stage of cellular respiration. So I understand your confusion because we use the term kind of loosely. Let me see if I can rephrase that. If you're breaking down glucose, glycolysis is the first stage, and then the pyruvic acid enters the mitochondria, and a whole bunch of other stuff happens in the mitochondria. Okay? If it's a fatty acid, you actually don't have to do glycolysis, and you don't make pyruvic acid. The fatty acids just go, I think, directly into the mitochondria and enter into some of the reactions there. But we still call that cellular respiration because it still includes the part where most of the ATP is made and where you use the oxygen. So I guess not all cellular respiration includes glycolysis, only if you're starting with glucose. So I'm not sure. That's kind of funny. That makes me think I'm not sure then how we define cellular respiration. It must be more defined by the part that happens, the part that we call the Krebs cycle. Marwa says we're not using lactic acid fermentation. You are actually doing a little bit all the time, kind of a trickle, but you don't do a lot until you have a sudden sort of need for more ATP in anaerobic conditions. Uh, okay. Yeah, that already got answered. I'm trying to skip a few things that you guys have already answered. Okay, Tanmay, that was a good description. You wrote lactic acid fermentation is only used when oxygen isn't available for muscle cells. Yes, that's a nice way to put it. Laura's asking a specific class question. Are we going to get our checkups back? Definitely. So the plan for the block day is that you'll get your checkups back. I can answer questions, and then we'll go over the practice exam. We're going to do another little exercise to help you with test taking, and then we'll take the test. You'll take the test. I'll watch you take the test. I have to grade your test. Okay, so Rohin, the short answer question is your model, right? A little bit different this time. Rest of the year you get FRQs. Enjoy the model. So Ankita's asking, do muscle cells also do respiration? Yes, if they have enough oxygen, right? So remember, lactic acid fermentation is sort of a stopgap when your muscle cells don't have a lot of oxygen. Okay, Kush, I already talked at some length about the connection between ATP and structure, so I'm going to let that one go right now. Okay, Laura's asking about somatic stem cells originating. So I haven't used the term somatic yet, but I'm going to start using it a lot when we get to genetics, okay? So get comfortable with that. Somatic just means most of our body cells that are not what's called our germ cells, like the reproductive cells that eventually can divide to form eggs and sperm. So I think the reason that's coming up is that the resource I gave you, the Inside the Cell, or maybe it was a website, I don't know. One of the resources calls what I think of as adult stem cells somatic stem cells. It just means like body stem cells, okay? And they still everything that's in your body came from your embryonic stem cells, okay? So some of them differentiate all the way into a very specialized cell, like a skin cell. Some of them sort of differentiate part way, right? Like they can't become everything, but they can still become a few things. But all of those originated, all the cells in your body originated from your embryonic stem cells. Caitlin's asking about a resting state. You're using about half and half carbs and fats. Mara's asking what type of fuel occurs in the cytoplasm. If you mean what process, that's where glycolysis happens. I'm not sure what you're referring to. Okay, Joseph is asking, does denaturation change the R groups of the amino acids themselves? No. So if you recall way back when we learned about proteins, there was this primary, secondary, tertiary structure. The primary structure being the sequence of the R groups. And then the secondary structure is where the chain starts to fold up and form these sort of regular patterns. Sometimes they kind of zigzag or form these patterns that have to do with these, I think, hydrogen bonds and disulfide bridges or something that you don't need to know, okay? But before they form their sort of special, unique shape, they form these sort of regular patterns. And the bonds that are holding these patterns together and the bonds that are holding these ultimate unique shapes together, that's what gets broken in denaturing. The R groups, the chain is still amino acid, amino acid, amino acid. It's just not folded in the same way. So no, the R groups themselves don't change. It's the bonding that is between the different amino acids that's causing it to fold up and sort of bind itself together. Minjay's asking about tips for studying. I think, a few things. I would say making sure that you can use specific examples in context, right? I'm not going to ask you, when I write questions, I want to try to make them as sort of realistic to what's happening in living things as possible. So I'm going to use specific terms like hemoglobin or lactase or whatever, and I'm going to put it in the context of general cell biology. So you want to be able to take a general concept and stick in different specific molecules that apply, right? So you might want to think in those terms. Could you describe enzyme function with the 2 or 3 different enzymes that you've learned about? Could you describe a complex thing like blood glucose regulation with all the different parts of that, the cell transport, right? The names of specific things. That's kind of what I'm trying to do in the model, right? Is to give you a context to use the general ideas, but with very specific terms. So as much as you can practice that, try to pick out some vocabulary terms and see if you can use them in context and see if that's helpful. Go back and forth between cell bio 1, 2, 3, and 4, right? Make sure that you can put all the pieces together. And I've written questions, I hope, that get you to do that, at least to some extent. I'm not sure who IT is. Your comment kind of made me smile. I'm sorry because it sounds a little stressful for you. You wrote that everything made sense. Then I read the practice exam. So try to determine why that is. Is it because the applying your knowledge to those questions was challenging, or is there some specific thing that I can help with? Okay, but otherwise, use that as sort of practice to think about how you can put things in context. Yes, have a positive mindset. I agree. We're actually going to practice some stuff around your mindset around test taking tomorrow or Thursday. Yes, Rohin, those short answers on the practice test were just for practice, right? Partly because it takes me a long time to write multiple choice questions. So, yeah, ID, you're not going to fail. At least I hope you don't. I don't know who you are, but I hope you don't. Okay, Liana's got some study suggestions here. Check your notes. Teach it. Absolutely. Teaching is the best way to learn. Do not read a ton of text. I agree. Make your study guide appealing so you don't get bored. Absolutely. Great ideas. I miss you, Liana. I hope you're having a good time. I can't remember whose class you even went to, but have a good time. I think you went to Miss Chow's class. She's pretty awesome, too. Okay. Okay. So Daniel's saying we're not using too much energy right now when we're just kind of hanging out. Yeah, it's related to the reason we're using both. I mean, if you think about it, we're not using energy at a rate where oxygen becomes a limiting factor. So that is part of the reason that we can kind of afford to use both. And we have more fat stored. So, if we don't need to use all our glycogen, and this is kind of me brainstorming a bit. I don't think our body's in a hurry to use up all our glycogen if we're just sitting here, right? Because part of the function of glycogen is to allow us to, quickly produce glucose for blood glucose regulation or for sudden movement, right? So it's sort of to our benefit to sort of use these longer term reserves, at least to some extent, if oxygen and time are not limiting factors. But it is, of course, a little more complicated than that. Different cell types do have to use different fuel, et cetera, et cetera. Thanks, Ankita. Yeah, you guys are asking really good questions. A lot of you are answering questions. You've learned so much in several weeks. Like, you should feel really good about everything you've learned. And I think if just be mellow, answer the questions, use good strategy, I think you're going to be fine. Especially those of you who are hanging out on this live stream. I think you're going to be fine, okay? Um, but yeah, this is challenging material. Okay, I don't know if it'll make you feel better or worse, Ankita, but for cell size and scale, so for everybody, I'll give you a hint about the test. You're not going to be sorting like a list of terms like I've done on the other assessments. I don't know, done enough of that, right? So there's actually a multiple choice question with like some comparisons. So you might want to go back to, there was a practice I gave you when we were practicing size and scale right before the cell biology quiz, where it compared some terms and you had to sort of put down your reasoning of why things were bigger than other things. That might be a good thing to just take a quick look at. There is a question that's sort of similar to that on the exam. Okay, there's a question about glycation. I've never heard of a word glycation. I'm wondering if you mean glycosylation, which has to do with glucose attaching to proteins. It's something that happens when people have high blood glucose. Not sure, so I don't know what that word means. Daniel's asking, do we need to state that water flows from high to low concentration, or can we just say that water follows solutes? I'm trying to keep it simple, so I'm okay with water follows solutes, as long as it makes sense in the context of what you're explaining, that there are solutes for the water to follow. Yeah, that's fine. Technically, of course, it does flow from high concentration to low. Okay, Joseph just gave you a fun example of some different molecules. Okay, Sharon, not sure I've got your name right. You're asking about how cancer has a bad impact on the body. I think sometimes it physically crowds and damages the other cells. My understanding is they put out some toxins. They steal resources from your other cells, and they stop other cells from, or reduce the number of cells doing their normal function, like acting as your liver or acting, in whatever cell function they have. And so there's a number of ways that they both damage tissue and put a lot of stress on your body, so that eventually it just becomes overwhelming, and you start to have your organs fail and that sort of thing, and then you might actually die from that. Ryan, regarding if other teachers are putting FRQs, I believe so. I'm the only one who does the models. I kind of made that up, and I really like it. Partly it's just a nice change of pace. We write a lot the rest of the year, but I do believe the other teachers are using FRQs. They tend to call them A-level questions, and I believe they're all using some kind of written one. Not sure if that matters, but yes, I believe they are. So Mara was asking, how do you know what is a limiting factor in any situation? I'm not quite sure. I think based on the questions you were asking in class, um, I guess you need to know the context, right? I mean, you would have to know some information to know what the limiting factor is, right? But you have enough background knowledge at this point to make some predictions, right? So we talked in class about how it's highly unlikely that carbon dioxide would ever be a limiting factor, but here in places like California that are dry, water is likely to be a limiting factor for plants. In the rainforest, it might be things like light or space or nutrients. Okay, so the idea of limiting factors is to sort of have a sense of the environment, the context, and then to consider what things might run out of first, or what might stop them from going further. Gia, yes, for the model, almost all those things should be showed with mostly diagrams. You will need some words, obviously. You want to label diagrams, right? Like you don't want your diagram to just be, sometimes if it's just pictorial, it's hard to tell what's happening. So for example, if you have an arrow, I need to know what thing is moving, right? So if you're trying to show the movement of water, maybe you'd want to write H2O or water, and maybe the word osmosis, right? So that it's clear what your picture is showing. That being said, if you just write water moves through osmosis, I don't really have a sense for sort of like, do you understand where it's moving? I think a picture is really good for that. So I would say sort of base it in pictures, but have enough words. And it's okay to have phrases to explain ideas, but because I'm trying to encourage you to have this sort of visual sense of all these little physical things happening, I want to see through the model that you can form a picture of these things happening, that you're not just sort of like spouting out a bunch of words. Like I want to see you can put the physicality of what's happening together with the words. So Ankita, the test is not the same as the practice exam. Again, those short answers don't exist on the test, but I tried to hit some of the concepts, and the model is exactly the same. Leo, the unit after cell bio is genetics. I'm excited. It's going to be fun. Is the practice test harder than the test? It's IT. I'm not sure which questions were worrying you so much on the practice test, and I don't have it in front of me right now. You know, the test is longer. Obviously, there's a lot more multiple choice questions. I don't know. I don't know if it's harder. I didn't compare them. You know, I just tried to write some questions that were similar. It's not intended to be dramatically easier or harder. Huh? Ananya's confirming she meant glycation. I don't know. I'd have to look that up. I've never heard that word before. Ananya's saying it was on the skeleton notes. That's probably something from another class, so I'm not quite sure what you're referring to. If you could give me some context, I could try to help. Ankita, another thing I've let go of, I always feel like I need to justify that. I know it feels like there's an awful lot of content, and there is, but I've let go of a lot. So a few years ago, I let go of hypertonic and hypotonic. They come up occasionally, but they're not required. They will not be on the test. Okay? But very briefly, the word root hyper means sort of like above a lot. So hypertonic is a high concentration of solutes relative to something else. Hypo means below, beneath, something like that, less. So hypotonic is a lower concentration of solutes compared to whatever you're comparing it to. Tenmei is just kind of asking ahead. We're actually going to start genetics on Friday. I know it's going to be a little weird timing, just kind of the way it worked out. So we're going to do genetics Friday. You'll be working on it the following week before Thanksgiving, and then we'll continue on until right before finals. Laura's asking about tutorial tomorrow. Yes, so tutorial tomorrow, like today, is going to be just focusing on exam review. Okay, so bring questions. Happy to cover that. And by the way, that's a good time to point out that I'm going to try to wrap this up soon. It's 9:30, so I want us all to make sure we get some rest. So yes, if we don't get your question answered tonight, feel free to drop by during tutorial tomorrow or ask during class. Okay, I'm going to skip all the comments about hypo, hyper. Daniel's saying a cell placed in hypertonic solutions lose water. Yes, the water would go out. Yes, that's correct. Okay, Leo, so let me clarify. So you said tomorrow's retake day at tutorial. No, tutorial is exam review. I will be doing retakes during lunch. Okay, so I'm not quite sure I'm going to balance it all, but I'm going to try to do retakes for a bunch of things during lunch. So that's going to be a little crazy. Okay, glad I got your name right, Jared. Yeah, I think vesicles probably are bigger than glycogen. Although glycogen, remember when we went over that briefly before the practice quiz, is a particularly large polymer. And vesicles probably can be different sizes. I'm not going to be asking you to compare things that are sort of in a gray area like that. So don't stress too much about that. They're going to be comparisons that should be obvious to you if you're prepared. Laura's asking, should you do the diagram first? Ultimately, I don't think it matters, to be honest. If you're worried about it, then do it first. But don't drag it out, right? There's not actually that much in the prompts, right? So, don't spend more than you should be able to do it in like 15 minutes. It really shouldn't be that extensive. You'd want to probably save a half an hour, maybe 35 minutes or something for the other questions. So, keep an eye on the time, but don't go too crazy with the diagram. I'm going to allow, depending on how long it takes us for the other things, maybe around 55 minutes or so for the test. That really should be plenty. So Leo, you're mentioning retakes. I think you might be talking about the cell bio quiz retakes. Those take a while, and I still have a lot of people. I may not get to them this week, realistically. I don't know. I may do some Friday, I'm not sure. Friday tutorial is kind of the last time for other things, so I would rather, frankly, do the quiz retakes later and finish everything else up, because I don't want to be dragging other stuff into next week. Okay, so Friday is the absolute deadline for quick quiz retakes, scored assignments, all that. And if I can't get to the quiz retakes, which I'm going to be honest, I got a pile of them, probably not going to happen, I will schedule a date for those next week. Krish, if you can't make tutorial, that's just review, so that's up to you. So I'm not sure what you want to come in for during brunch or lunch. I would say not during brunch. Lunch, you can come in for retakes. Mara was asking about the optional textbook questions. I'd skim, if you have time, you want to skim them, but it's getting late. I wouldn't stress over it. It's more just for quick recall. A lot of review is really about seeing, do I know this or do I still need to study this, right? It's not that those particular questions are going to be related to the exam. I honestly haven't looked at them in a long time. It's more just like, do I remember this content? I suspect you do, so I'm not too worried. Okay, not sure about your question, Marwa. If you understand concepts will be okay, or do you recommend us to know scientific or specific information? So remember what I said. You do want to put specifics in the context of the bigger concepts, right? So not just active transport and facilitated diffusion, but how does that relate to blood glucose regulation or maintaining the pH of a lysosome? So yeah, you want some specifics, but you want to be able to fit them into the context. And yes, Divya, I mentioned that before, but we are going to debrief the practice test before the exam. Okay, Ankita, try not to stress about the diagram. It really is not meant to be overly complicated. Okay, if you look at each of the prompts, there's certain, very specific things I'm looking for in terms of, connecting. If you can show how the stomata work, if you can make the connection to structure and function that was in the cactus question, right? So I'm looking for, kind of very particular things of how those molecules are used. And they're all honestly pretty basic. It's not like obscure stuff. It's stuff that we spent a lot of time on. So I think you'll be okay. Again, the biggest single thing people do wrong on the diagrams is just not answering all the prompts. So just check them all off as you go. The diagram is, it's 20 points. I've been trying to be consistent about making FRQs, and the diagram is kind of an FRQ, about 20% of the overall test. So like they're significant, but not overwhelming. I'm kind of curious, IT. It sounds like you're in my class, but I'm trying to think if I know of anybody with the initials IT. I don't know. If you feel like, let me know. Be nice to know who you are. Sharon is asking, what was the second process of cellular respiration? Not sure I understand the second process part of your question. Lucas, no, we did model review in class today. We're not going over it again. Okay, it just would take too long. So hopefully you guys had, some time to talk in class today. We're just going to go over the question part of the practice exam. Kush, I'm going to be way busy with retakes during lunch. So I would say if you have a question, ask a friend, ask them to ask me during tutorial, something like that. Because no, I'm going to have like a gajillion people there for retakes during lunchtime. Joseph, is lactic acid fermentation used by all eukaryotes? I don't know. It came up earlier, Joseph, about whether plants do fermentation, and I've never heard of that. So I think the answer is no. Again, sometimes I don't know stuff, but I don't think plants do lactic acid fermentation. And I don't know if fungi does. Oh, wait. Yeah, I mean, yeast do alcoholic. I don't even know if fungi can do lactic acid fermentation. Laura, I did answer that question at some length about structure and function. I guess briefly I'll say, look at your cactus. The one about plant structure and function that had the cactus example on the back, that kind of puts together a lot of the plant stuff you need about structure and function. So I'll refer you back to that. You're welcome, Ankita. You're going to do fine. I really appreciate all your hard work. You have a lot of really good strategies, okay? So feel good about what you accomplished. Okay, so I think we're gonna call it a night here. Okay, yeah, you guys really relax. I'm not too worried. If it helps you to hear this, I'm happy to tell you that students do generally somewhat better on the cell bio exam compared to the cell bio quiz. Okay, because there's more questions, you sort of have more opportunities to show what you know, and usually you've had more time to put it together in your head. Okay, so I think you're going to be okay, right? Just focus on being okay mentally, getting some good sleep tonight, thinking clearly during the test, using good test strategy, okay? And do your best, okay? It's going to be fine. So I'm going to leave it at that, and I hope you guys have a wonderful night. Again, don't stay up too late, get some good rest, and I will see you all, well, not all of you, it's a block day tomorrow. I will see you all either tomorrow or Thursday. So have a wonderful evening, and hope you enjoyed this BioLerner presentation review. Take care. Have a good night.